Before we dive into the complex world of DNA and heredity, let's travel back in time to a quiet monastery garden in the 19th century. Here, a monk named Gregor Mendel, through his meticulous work with pea plants, laid the foundation for modern genetics. He didn't know about DNA or chromosomes, but he observed patterns in how traits were passed down from one generation to the next. These observations led him to formulate two fundamental principles, starting with what we now call the law of segregation. To truly grasp this law, we first need to understand some key terms. Imagine your body is a massive library, and every book is a chapter on how to build and run you. Each of these books is a chromosome. On these chromosomes are specific instructions, or recipes, called genes. A gene might be the recipe for your eye color, your hair texture, or whether your earlobes are attached or detached. Now, for many genes, you don't just have one recipe, you have two, one inherited from each of your parents. These different versions of the same gene are called alleles. Let's use the example of pea plant height, just like Mendel did. There's an allele for being tall and an allele for being short. If an organism has two identical alleles for a trait, say, two tall alleles, we call it homozygous. If it has two different alleles, one for tall and one for short, it's called heterozygous. This is where the concepts of dominant and recessive come in. A dominant allele, which we represent with a capital letter like T for tall, is like a loud voice in a conversation. If it's present, it's the one you hear. The recessive allele, represented by a lowercase letter like T for short, is the quiet voice. It's only heard if both alleles are the quiet recessive type. So, a plant with the alleles TT, homozygous dominant, will be tall, and a plant with TT, heterozygous, will also be tall, because the T allele masks the T. Only a plant with TT, homozygous recessive, will be short. The combination of alleles an organism has, like teat, is its genotype. The physical trait that you see, like being tall, is its phenotype. With that foundation, we can now properly introduce Mendel's first law. The law of segregation states that for any given trait, an individual's two alleles separate or segregate from each other during the formation of reproductive cells, which we call gametes, sperm in males and eggs in females. This means that while the parent organism is diploid, meaning it has two sets of chromosomes and therefore two alleles for each gene, the gametes it produces are haploid. A haploid cell contains only one set of chromosomes. As a result, each gamete carries only one allele for each gene. This elegant process ensures that when two gametes combine during fertilization, the offspring will once again have two alleles, restoring the diploid state, but with a new, unique combination inherited from its parents. Core principles. The mechanism. So, where does this masterful sorting happen? The biological magic behind segregation occurs during a special type of cell division called meiosis. Specifically, it happens in a phase known as anaphase 1. During this stage, the pairs of homologous chromosomes, the ones carrying the different alleles from each parent, are pulled apart and moved to opposite ends of the cell. This physical separation is the very essence of segregation. It guarantees that the resulting daughter cells, which will eventually become gametes, each receive only one chromosome from each pair, and therefore, only one of the two alleles for every gene. Random selection. Crucially, this separation is a game of chance. The allele a gamete receives is determined completely at random. Think of a heterozygous parent with the genotype AA. When this parent produces gametes, which allele goes into which sperm or egg cell is like a coin flip. There is a 50% probability that a gamete will receive the dominant A allele, and a 50% probability that it will receive the recessive A allele. The parent doesn't get to choose. This randomness is a cornerstone of genetic diversity. Restoration at fertilization. The process comes full circle at fertilization. When two gametes, one sperm and one egg, fuse to create a zygote, the diploid number of chromosomes is restored. The resulting offspring now has two alleles for the trait again, one contributed by the maternal gamete and one by the paternal gamete. This combination of alleles, its new genotype, will determine its physical characteristics, or phenotype. The purity of gametes. This is why the law of segregation is sometimes poetically called the law of purity of gametes. Before Mendel, it was believed that parental traits blended together in the offspring, like mixing paint. But Mendel showed this wasn't true. A gamete is pure because it can only carry one allele for a specific trait, either the tall allele or the short allele, but never a blend of the two. The alleles remain distinct, individual units. Classic example: Mendel's pea plants. Mendel's genius was in his experimental design. He started with what he called true breeding or pure breeding plants. These were plants that, when self-pollinated, always produced offspring identical to themselves. He took pure breeding tall plants, which we now know had the genotype TT, and crossed them with pure breeding short plants with the genotype TT. This initial cross of purebred parents is called the P, or parental generation, F1 generation. The results of this first cross were striking. Every single one of the offspring, known as the F1 or first filial generation, was tall. The short trait had seemingly vanished. But Mendel knew better. He understood that each F1 plant had inherited one allele from each parent, giving them all a heterozygous genotype of TT. Because the tall allele T is dominant, it completely masked the presence of the recessive short allele T. F2 generation. Here's where it gets really interesting. Mendel then allowed these F1 generation plants, TT, to self-pollinate. According to the law of segregation, each F1 plant would produce two types of gametes in equal numbers, half carrying the T allele and half carrying the T allele. When these gametes combined randomly, the hidden short trait dramatically reappeared in the next generation, the F2 generation, making up about 1/4, or 25%, of the offspring. The 3:1 ratio. This experiment, when repeated hundreds of times, consistently produced a phenotypic ratio of approximately 3 tall plants for every 1 short plant. This iconic 3-to-1 ratio was the critical evidence for segregation. It proved that the short factor hadn't been blended, diluted, or destroyed in the F1 generation. It had simply been segregated during gamete formation and then recombined in the F2 generation, ready to express itself again in a homozygous recessive TT individual. This simple observation shattered the old idea of blended inheritance and gave us our first real rulebook for heredity. Now, let's build on that foundation with Mendel's second law, the law of independent assortment. This law takes things a step further. It addresses what happens when we track more than one trait at a time. It states that the alleles of 2 or more different genes get sorted into gametes independently of one another. In simpler terms, the allele a gamete receives for one gene, say plant height, does not influence the allele it receives for another gene, like seed color. It's like shuffling 2 separate decks of cards at the same time. The outcome of one shuffle has no bearing on the outcome of the other. The mechanism. The biological basis for this law is also found in meiosis thought, but this time in a stage called metaphase Y. During metaphase Y, the homologous chromosome pairs line up at the cell's equator, called the metaphase plate, just before they are pulled apart. The key here is that the orientation of each pair is completely random and independent of all the other pairs. A chromosome pair carrying the genes for seed color might have the maternal copy on the left and the paternal on the right, while the pair for flower color might have the paternal on the left and maternal on the right. This random lineup creates a massive number of possible chromosome combinations in the resulting gametes. Mendel's P experiment. To discover this, Mendel performed what are known as dihybrid crosses, which means he tracked 2 different traits at once. For a classic example, he crossed a plant that was purebred for 2 dominant traits: yellow, round seeds, with the genotype YYRR, with a plant that was purebred for two recessive traits, green, wrinkled seeds, YIRR. Just as before, the F1 generation was uniform. All offspring had the genotype YR and displayed the dominant phenotypes, yellow, round seeds. The real test came when he self-pollinated these F1 plants. Because of independent assortment, a YR plant produces 4 different types of gametes in equal proportion: YR, 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 and YR. When these 4 gamete types from both parents combine in every possible way, it results in the famous 9:3.3.1 phenotypic ratio in the F2 generation. That is, 9 offspring showing both dominant traits, yellow and round, 3 showing 1 dominant and 1 recessive, yellow and wrinkled, 3 showing the other combination, green and round, and just 1 showing both recessive traits, green and wrinkled. This precise ratio could only be explained if the genes for seed color and seed shape were being inherited independently. Importance. Why does this matter so much? Independent assortment is a primary engine of genetic variation in sexually reproducing organisms. It shuffles the genetic deck with every generation, creating new combinations of traits. This variation is the raw material for evolution. In humans, with our 23 pairs of chromosomes, the random assortment during metaphase I can produce over 8 million different genetic combinations in our gametes from this process alone. That's 2 to the power of 23. When you then consider the random fusion of gametes from 2 parents, the number of unique genetic possibilities for an offspring skyrockets into the trillions. This is why, unless you're an identical twin, there's no one else on Earth quite like you. Exceptions. Linkage. Now, it's important to note that Mendel's second law comes with a major caveat. It only holds true for genes that are located on different chromosomes, or for genes that are very far apart on the same chromosome. Genes that are physically close to each other on the same chromosome are said to be linked. Because they are on the same piece of DNA, they tend to be inherited together as a single unit, violating the principle of independent assortment. They don't shuffle independently because they are, quite literally, stuck together. The study of this phenomenon, called genetic linkage, opened up a whole new chapter in genetics that helped scientists map the locations of genes on chromosomes. But for the vast number of genes that do assort independently, Mendel's law remains a powerful predictive tool.